We'll start. So today's class is on periodontal disease activity models. Okay, so when a patient comes to our clinic and we found out that this patient is having a periodontal disease, and we tell the patient that you are having periodontal disease and for which you may have to even extract some tooth. The patient may be on a shock and asking that I didn't even notice such a thing was occurring to me. Okay, so how to deal with these type of patients? We can say that it may be due to the local factors that is present, or it is due to some hereditary factors, or it is due to the age-related factors. Like that, the reason goes on. But how much we can convince the patient? Okay. So for that, we should know what was our basic what research till now, what is occurring. So that is what today's class is about. That is periodontal disease activity models. From the historical, from the start of the history, how we can up to now, what is the knowledge that we have gained about the disease activity of a periodontal disease. Okay, so that is, first we'll say that it is due to the local factors. Hereditary, it may be due to the hereditary or due to the age-related factors. So we'll get to know that from the history to the current understanding of the periodontal disease activity. Now, when come to the history, there are certain models, that is continuous model, asynchronous multiple model, rapid burst model. That is a historical point of view. Now, there are a lot of theories that have explained about the disease activity. Now, these three are the most important. That is what I have just included in the slide. Now, recently, we have the non-linear model. That is by the Kahneman, Kahneman model that we have very well accepted. Then the contemporary model of the host microbe interaction. Now, and also I have included what are the current concept, that is mainly about the keystone pathogen concept, and polymicrobial synergy and dysbiosis model, which has included the keystone pathogen concept, and a little bit about the genetic dysbiosis concept, which was formed in 2015. So these are the basic models that of periodontal disease activity. Now, the first one is a continuous model. Now, this model was formed prior to 1980. So this is considered to be one of the model that from the historical point of view, the first one to be we are understanding. So according to this, the periodontal disease activity is a slow and continuous process. That is, it is the affected site is with constant progressive rate of destruction. So when we draw a chart, draw a graph with the time and the disease of the rate of disease, we can almost draw like a horizontal line. That is, with the age, it is the disease is progressing. Now, throughout the duration of the disease, we can see this. So what is the inference from that? That is, at the end of the, as the disease increases, all the gingivitis cases will progress to periodontitis. Okay, so that is what they had presumed that. Now, when we see the graphical representation, it will be like this. That is, in the disease progression, we have the y-axis, and the time we have the x-axis, and we can see a horizontal a line, not horizontal, a tangential line that is of a constant rate. Now, according to this model, it was believed that all the cases of untreated gingivitis will lead to periodontitis. Now, we very well know that this is not the situation that is occurring. There are cases at the age itself, it still persists as gingivitis. All the cases may not progress to periodontitis. So this model actually has the drawback of this limitation. Now, what is the next model? This is again a graphical representation of the continuous model. Here you can see that in each side, the first one is the timeline, the second line is the side. So according to each side, how it is different. Say for example, the first one maybe of 1 1, the second or third one maybe of 1 4, or like that. So anyway, in 1 1, according to the time, there is a constant increase in the disease activity. In the 1 4 also, with the chronological period, there is a constant increase in the disease activity. So there is no concept of period of remission, or there is no concept of inconstant progression of the disease activity. Okay, so the next model is asynchronous multiple burst model. Okay, so the first one was continuous model. The second one is asynchronous multiple burst model. Now, according to this, the periodontal disease activity occurs around the affected tooth during defined periods of life. Okay, during the defined periods of life. Okay, so at a certain period of life, there is a disease activity which is occurring in all the tooth that is having a disease chance. Okay, at that time, at all the teeth, it is having the periodontal disease activity. Okay, so after that, there is a burst of inactivity or remission. Again, there will be a defined period of life in which there is a disease activity. So one thing that we have to know that in this multiple burst asynchronous model, at that they have specified the term that defined period of time. So at that time, all the teeth is having the periodontal disease activity. Okay, so the chronology of these bursts of disease is asynchronous for individual teeth or group of teeth. Now, when we plot a chart or when we plot a graph, we can see that in the one, for example, we had given it as 1 1, 1 4, 1 6, and 1 7, right? So in 1 1 or 1 4 or 1 6, 1 7, the disease activity period is same in all the tooth. Okay, so at that time, there is disease activity in all the tooth. So note the point of note the word that is defined period of time. Now, the next model, the next historical model was rapid burst model or episodic model. Okay, so in this, the change is that there is a short burst of destruction which is followed by a period of no destruction. Now, what is the difference in this model is that they have not mentioned a term like a defined period of time. Okay, so in each tooth can differ. In the previous example, we had told that in 1 1, if that tooth is having a disease activity, at that time maybe the 1 4 may not have the disease activity. Okay, so the pattern is random with respect to the tooth sites affected and chronology of the disease progress. So it has no relationship with the chronology, and also it has no relationship with the tooth site. Okay, so that is rapid burst model or episodic model. Now, how can we draw the graph here? When we draw the timeline and the sites, we can see that the 1 1 or 1 4 is having different disease activity period. Okay, different disease activity period. When there is disease activity in 1 1, there won't be disease activity. There may or may not have disease activation at the 1 4 site. So it is completely random. It is the periodontal disease activity is completely random. So that was a 3 historical model, which is actually some sort of acceptable. That is a continuous model. Then the second one was the rapid asynchronous multiple burst model, and the third one is the rapid burst or episodic model. This was the historical part. Now, coming to the recent part, we have the non-linear model. We very well know that the Conman's concept of disease activity. You have the 4 tables. You have the disease activity. You have the host response. You have the clinical connective tissue and the bone destruction pattern and the disease expression. So that 4 tables. That is the main presentation of the non-linear model. This is actually one of the most acceptable one of model of the disease activity that has put forward by Kenneth S. Coleman in 1997. Okay, so according to this, it is in this concept of model they have mentioned the term of the interaction between the host and the microorganism. So it is in this model you have the host response model, and here in this model they have also considered the clinical expressions that depends on the genetic and environmental factors that modify the host response. Now this is the 4 tables that we have mentioned, that is the microbial challenge, the host immune inflammatory response, and the connective tissue and the bone metabolism, and the clinical signs. And the uppermost part we have genetic risk factors, and the lower side they have considered the environmental and acquired risk factors. Okay, so now when coming to in 2008, Coleman again modified this non-linear model to multi-level hierarchical and biological system of models, in which they have came up to the level of micromolecular level, that is the interactions reflected in the gene, protein, and the metabolic expression patterns. Okay, so these genetic and environmental factors, when it has so and so actions, how it is acting up to the gene, protein, and metabolic expression pattern. Okay, up to that level, they have included in the multi-level hierarchical and biological system of model by Coleman, again by Coleman in 2008. Okay, so this is the basic model of the non-linear model. Now, the next one is the biological system model. Oh, okay, the one point I have been describing about the historical point. Now the recent models, the first one was the Coleman's model, and the Coleman's model, the latest model I have included. Now next is the biological system model, that is by Offenbacher. Offenbacher, we very well know that he's a famous scientist who has involved with periodontal medicine, that too many researchers on preterm lower babies and the relationship between the periodontal disease. Now, according to this model, that is the biological system model, we know that when a patient asks to you that when a patient comes to us with diabetes and another patient with non-diabetes, the pattern of disease activity of periodontal disease is different. Now, how it is acting? A diabetic patient, how the disease progression is acting, and non-diabetic patient, how the disease progression is acting. So in the the first one is the personal level. The first circle they have included the personal level, that is the biofilm characteristic, and the environmental factors that including the smoking, diabetes, obesity. If the patient is having the risk factors, then their presentation of disease activity is different. Now, when this patient is having to the next level, that is the genetic level, if the patient is having a genetic polymorphism or epigenetic modification, along with the risk factors, again, the disease pattern differs. Okay, now these cumulative factors will depend on the biologic phenotype expressions that is expressed in the third ring. That is, the biologic phenotype expression means the host immune inflammatory response, the cellular molecular changes, and the inflammatory biomarkers. Now, all these 3 factors will finally express the clinical phenotype presentation of the disease. Okay, so mainly there are 4 levels, that is the person level, genetic level, biological phenotype level, and the clinical presentation. How the clinical presentation of disease is differing in each and every individual. This is the model that is by Offenbacher. Now, again, there is more explanations needed in this model also. So, scientists have came up with another model, another concepts. Actually, model as such, it is not now. Now, they have also came up with certain other models too. Now, this is another model that is a contemporary model of host-microbe interaction. That is by Bailey and Chappell in 2015. Now, this is the 2015. This is one of the model that is most recent concept. And in this model, they have considered clinical health. That is the difference in this model. That is, we till now they have explained that there is gingivitis, there is periodontitis. We have to consider also clinical health. Health means it is not the meaning that there is zero zero microbe host response. There is microbe, there is host response, but it is in the balance. That is what is happening in clinical health. Now that concept they have introduced in this contemporary model. So in the first figure, you have the clinical health, in which the health promoting biofilm symbiosis is occurring. I hope we know what is the term of symbiosis. That is a microbe, different microbes living together. Okay, that is symbiosis. Now, when that is stable, the host response is proportionate. Host response is proportionate. Now, when gingivitis, when we tell it as gingivitis, incipient dysbiosis occurs. That is, the symbiosis is disrupted. Certain microbes will become pathogenic. Okay, they will proliferate. They will rule that that part now. Okay, so this dysbiosis has occurred, but still the host response is good. So again, you have a proportionate host response. You don't have a disproportionate host response that will occur to more destructive patterns. So now it is in the gingivitis stage itself. Again, there are a lot of factors that is determining. That is the behavioral factors like the habits, and the environmental factors like smoking and other risk factors. Now, when there is frank dysbiosis, that is when the dysbiosis is to the much level, and when there is disproportionate host response, that will occur more and more of connective tissue and bone damage, which will cause a established periodontitis. Okay, so that is the basic model of the contemporary model by Neely and Chappell in 2015. Now, what are the current concept of disease activity? We have heard about the keystone pathogen concept. That is, Porphyromonas gingivalis is considered to be one of the keystone pathogen. Now, what is the relevance of that? Even though when there is a symbiosis, if there is a dyssymbiosis, certain organisms have just mentioned can rule apart. They will be dominating. Even though their number is in a small number, they can actually immunomodulate other microorganisms to become more pathogenic, and it will cause the more worsening of the disease. That is the main keystone pathogen concept. Now, this keystone pathogen concept is included in the model that is known as the polymicrobial synergy and dyssymbiosis model by Higashi in 2012. Okay, so this is also one of the famous model, that is the polymicrobial synergy and dyssymbiosis model by Higashi in 2012. Now, in this, you have the health, in which there is symbiosis. When the dyssymbiosis occurs, the keystone pathogen will take the role, and it will cause immunoregulative mechanism, subversive mechanism, in which will cause more immune response to occur, and with which the connective tissue and the bone tissue destruction will occur more. Okay, so that is the polymicrobial synergy and dyssymbiosis model. Okay, now this is again the figure that is showing the symbiotic microbiota, and when there is dyssymbiotic microbiota, what changes are occurring and how it will lead to the periodontal disease. Now, this is one of the, again, the recent concept, that is genetic dyssymbiosis. This is again concept, not the model. Genetic dysbiosis. This is by Neely. This is by Steen and Caridis in 2015. Steen and Caridis in 2015. Now, what is the main concept is that the periodontal disease, why it has occurred, it is due to the genetic defect in the recognition and response pathway that the host uses to identify the microbial pathogens. Okay, when the pathogen comes to the when the pathogen comes to the gingival sulcus, the host should respond to it. So when there is a genetic defect in the recognition and the response pathway, the periodontal, there is an alteration in the microbial colonization, and also there is a misrecognition of the normal microbiota. So the host response will be different. Now, this genetic dysbiosis is the key concept of a term known as infectogenomics. That is a study that is mainly based on genetics and its relation with the disease activity. So this genetic defects, they have included in two main headings, that is the pathogen recognition defects and the bacterial proliferation defects. Okay? So those are the major models of periodontal disease activity. Now again, is it this the final disease activity model? Still we have to say it as no, because we very well know that in each tooth it is different, each site it is different. Now what is that factor that is what determining the change in each site? That area again we have to explore. Now again, whether there is the role of viruses in the periodontal disease activity, that also we have to know much more about. So our science, as we know, it keeps on updating more depending upon the research what we have done till now. So these query will be again changing. These what concepts will be again changing till we get an acceptable model. Now, for the time being, we very well know that we are very much sort of have a clear idea about the host response by the Kahneman's concept. So something about the host response also have the Kahneman has included. So this concept can again change, and we should have that mind to have that change also. Now, when coming to the first question, what was it? So when the patient comes to you, then how I got this disease? Now, again, we may have to tell that it may be due to the local factors, it may be due to the genetic factors, it may be due to the age factors. But now when we say that we have understood what is the history, what all research we have gone through, and why we have to tell these terms, why we are telling it as there is age, why we are mentioning it as genetics, and why we are mentioning it as local factors. Maybe on the next point, we may have to add certain factors too. Okay. So the best way is that we have to explain these are the factors to the patient, and we have to understand that this is the situation that is occurring. And since the periodontal disease, much symptoms won't be occurring, we may have to convince the patient more on that. Once the patient gets an idea, they may have the mind to accept what they have gone through, and which may help for the patient motivation and for next supportive periodontal therapy to after the periodontal therapy. Okay, so these are the various models of disease activity. Now, certain things is on the recent concept and certain is on the historical concept. So mix together and know about what is the periodontal disease activity. Okay.